Hello. So happy Friday, everybody. Welcome to this edition of New Frontiers in Spatial Omics, sponsored by the U.S. Humo Group. I'm Peggy Angel.
And I'm Kristen Burnham Johnson.
So for today's topic, we're going to be talking about integrative single-cell proteomics. So a current challenge right now in spatial omics is we're generating huge amounts of data, and we need to understand how to access that data to really uncover this spatial biology that we need to understand disease. So the focus of today's session is on experimental design and data pipeline applied to understand hepatic function and liver zonation.
And for our agenda today, we'll have two presentations from our speakers, followed by interview and then audience discussion as we normally do. And it's my pleasure to introduce our first speaker, which is Florian Rosenberger. And he is a European Molecular Biology Organization, or EMBO, postdoctoral fellow. He's working with Professor Matthias Mann at the Max Planck Institute of Biochemistry in Martinstreet, Germany. He holds a PhD in mitochondrial biology from the Karolinska Institute. Florian's research in the field of MS-based single-cell proteomics centers around understanding the metabolic niches in both healthy and disease contexts. So his work primarily focuses on liver biology and encompassing the development of single-cell proteomics maps to unravel the spatial-dependent proteome of hepatocytes.
And joining Dr. Rosenberger will be Caroline Weiss. So Caroline is a PhD student in the laboratory of Matthias Mann at the Max Planck Institute of Biochemistry. Her research interest focuses on combining spatial and single-cell mass spectrometry-based proteomics to investigate human physiology. Caroline's current work focuses on spatially dependent proteome characteristics of the human liver, aiming to deepen our understanding of liver zonation and its implications for disease and therapy. So, Florian, if you want to start, that's
Absolutely. Okay, wait. Can you see anything? Okay.
Yes, it looks like, there you go. It's in speaker mode.
Perfect now.
Thank you very much. Thank you, Christine. Thank you, Peggy, for the kind introduction. And thank you very much for having us here today. Also, everyone joining in, thanks for being here. My name is Florian. I'm here with Caroline. We're working in the lab of Professor Matthias Mann. The lab is known for technological developments in the field of mass spectrometry-based proteomics. And one of the hot forefronts at the moment is spatial single-cell mass spectrometry. What we're going to do in our talk today is we're going to tell you a little bit about our thoughts on this topic. In the first part, I'm going to go through some data that is already published. I'll talk about experimental design concerning a liver tissue single-cell proteomics. And then in the second part, Caroline is going to take you into the workshop. So she's going to tell you about an ongoing project, how she thinks about the experimental design, and what analysis pipelines she's developing for intact liver tissue. But before we do that, let's zoom out for a second. And as this is a spatial omics seminar series, let's just briefly ask, why is spatially actually so important? We're in Munich. Munich is known for its beer gardens and outdoor pubs here. This is generated with DALL-E, but this is very much how it looks like. The two of us are sitting inside. We didn't get any space outside there. So if you're a beer garden owner, it's extremely crucial where your pub is. On this busy square, you might have a lot of people around, but you also have access to resources. If you're unfortunate enough to open your beer garden in the Arctic regions, you have a problem because there are not enough resources to drive your pub and there are not enough people, although it seems like here are some. So let's transfer this metaphor to single cell or to spatial proteomics. So it very much depends on where a cell is to what it can do. So a particular cell might only have a particular set of enzymes around that are able to cope with the nutrient environment that is there. Like a liver cell would not be able to survive in the brain because the nutrients around it, the oxygen concentrations are not suitable. Very much like the beer garden example here. And if you might notice down here, these guys here have nothing on the table. So you also have to think about what are the individual entities that you're looking at. In this metaphor, it's the individual tables, but in single cell proteomics, obviously, it is the single cell part because the individual single cell is the entity that drives the function of the whole tissue. This is why spatial is so important. And it's actually a good time to do single cell and spatial proteomics with a mass spec. Why is that? So you've all heard about spatial transcriptomics. The proteins, the proteome that summarizes the proteins are actually the guys doing the job. And we and many others have observed that the proteome itself, the levels of the proteins are much more stable across cells than the transcripts are. This so-called stable coproteome enables us to measure a lot fewer cells than what we would have to measure with transcriptomics to get a stable signal. But also there is much more biology in the proteome itself than there is in a transcriptomics data set. Then secondly, if you compare the single cell proteomics with the mass spec to, for instance, immunostaining, we are able to quantify a lot more proteins at the very same time. So in this graph here, you see the development of bulk proteomics in the black line. This is not input limited, and on the y-axis here is the number of proteins identified, and we're beyond 10,000 proteins that we can quantify in any given sample. We think that any given cell type has around 15,000 proteins that we want to quantify. Single cell proteomics down here is a very young field, and it's just picking up. We are passing 2,000 proteins. Now we're up to almost scratching at 5,000 on a routine basis. So we're getting there. It's exciting times. But when you actually design your experiment, your single cell proteomics experiment, you have to think about the cell type that you're looking at. So cells have different sizes, very big ones here, like the oocytes or cardiomyocytes, but also extremely small ones like lymphocytes. The smaller a cell, the less protein it will have, and the more of an analytical challenge you will have to condense enough biology from these cells there. Single cell proteomics is a hot field, and many people think about this. If you want to get more into the concepts of this, I recommend reading the articles or the perspective articles in this focus issue of Nature Methods. Came out in March 2023. We also contributed one that is linked here via this URL and the QR code. But there are many, many more articles in there that are really exciting to read. Highly recommend this. And our mission now is to make single cell proteomics biologically relevant. Go from analytics to real biology. Here's how we think now, how we get the spatial context together with single cell proteomics. So obviously you have to somehow analyze your tissue. And at that time, the two postdocs, Andreas Munde and Fabian Koscher, in Matthias Mann's lab, developed a technological platform termed Deep Visual Proteomics, or DVP. This platform starts with any given tissue sample, for instance, archived clinical samples. We take very thin sections, typically 3 to 10 micrometers, stain those, so classical immunostaining, and then put them under a microscope. We then identify the outlines of cells, every single cell that is on there, and apply machine learning to identify the cells that we are particularly interested in. We then use a laser microdissection microscope to cut out these cells, sort them into a 384 well plate underneath the original tissue, and then we prepare our samples. This means going from proteins to peptides, and inject these peptides onto a liquid chromatography system that then goes into a ultra-high sensitive mass spectrometer. And then downstream, we do bioinformatic analysis. Caroline is going to talk about this a lot today, and we have the happy researchers and clinicians at the end of the day. This technology, with combining microscopy, machine learning on images, and mass spec, is extremely powerful. And now many people in the lab of Matthias Mann are working on this. It takes a village, really, and each one of us has their favorite organ or a particular disease that we're working on. You can see Caroline and me, we are here on the liver side, but we cover almost all other organs too. Just to give you an example how this works, this is work by Lisa Schweitzer, a former PhD student in the lab. She applied DVP to the fallopian tube. In there, you find two different kinds of epithelial cells, some that have FOXJ1 positive nuclei and others that have FOXJ1 negative nuclei. You can also see the cell outline marker APCAM that outlines every epithelial cell here. We then use machine learning to distinguish these two classes based on the FOXJ1 signal, is it positive or negative, and then cut out these cells via laser microdissection. I just start the video again. You can see here, this is at the beginning, it was a rather slow process. We are much faster now, but you can nicely see how the individual cells are cut out. There are two different ways. Either we cut out cells and pool them together, or we cut them out individually, which we're going to talk about later. Just as a proof of concept here, we can nicely separate the FOXJ1 negative and positive population on a principal component analysis that is a data reduction tool. And you can also appreciate that the FOXJ1 negative cells really have no detected FOXJ1 signal. So although these cells are directly neighboring each other, we are able to separate them very nicely with the laser. So when I came to the lab in 2021, the question was out there, can we apply DVP now to single cells actually in intact organs? So what has been done up to then was to cut out cells individually, but sort them together into a well. The question is, how can we cut out one cell at a time and measure it one cell at a time? And here I would like to go back to a graph that I showed you at the beginning, which is the cell size is absolutely crucial to how much signal you will get. So for this experiment to work, you need to choose a good model. We decided to settle on hepatocytes because they're a good mid-range model. There's a lot of known liver biology there too. And the liver is great for spatial proteomics and for benchmarking because the liver proteome is space dependent. Here on the right, you see a mouse liver section. We're staining for two markers, ecathirin in orange and glutamine synthetase in turquoise. And these two markers distinguish two areas in the liver: the portal vein areas where blood comes in, then gets drained through linings of hepatocytes to the central vein areas where the oxygen levels are lower and the nutrient levels have changed. This axis here, or this phenomenon, is called zonation, and this axis here is repeating throughout the liver over and over again. And here in this great review from the Porat Schlom lab, which we're also collaborating now with, you can also appreciate that there are a number of metabolic pathways that are highly up at one side of the liver, but down at the other side of this zonation axis, which we call the portal to central vein axis here. So how did we now go about with our spatial single-cell proteomics pipelines to map this zonation gradient here that we can so nicely see with immunostainings? First part was to establish a staining that would allow us to identify the outlines of cells, which we do with phalloidin. Then we mark the portal vein with ecathirin, and we mark the central vein with glutamine synthetase. And we have also a marker for both nuclei and the so-called sinusoid architecture. This is the streets, so to speak, where blood flows. Once the staining was established, we ran through this whole pipeline here of what we call single-cell deep visual proteomics. We took frozen mouse liver sections at 10 micrometer thickness, stained them, imaged them on a high content microscope, applied our AI-based cell segmentation to identify the outlines of cells, and then cut them out one at a time, sorting them into a 384 well plate underneath one at a time. In this particular experiment, we also used a labeling strategy called multiplex DIA or MDIA that is also published in a different paper. This allowed us to boost the number of proteins that we can reliably quantify. And downstream, use a timsTOF SCP mass spectrometer to get to our data. So how does the data look like? I told you at the beginning, the number of proteins is always extremely crucial in a proteomics experiment. We get on median 1,700 proteins from a slice of a cell. So we had 3 replicates. It dropped a little bit throughout this time. we cover about 3 orders of intensity magnitudes, including a number of transcription factors here, which are good to see because they are this is both intact biology, but they're also at the lower end difficult to catch. You can see that they're enriched down here. But what we also saw was that the area of the cell that we cut out correlated with the number of proteins that we would quantify. So you can see here, the smaller the shape, the less proteins we get, indicating that we're operating the mass spec at the lower end of sensitivity. And this is going to be important later in the talk again too. Do mind, this is only a slice of a cell that we're looking at here, because this is 10 micrometers. It's about 1/3 to half of a complete hepatocyte. And you can also see this when you actually look at the images. So here we have some cells where we have the nuclei, either 1 or 2. Hepatocytes can be binucleated. And these are those cells that have the highest number or the highest intensity in histone proteins, versus those cells down here that don't have a visible nucleus. They have the lowest intensity in histone proteins. So very nice confirmation. Now, what can we do with this data? So obviously we have the single cell proteomes. We know exactly at which position we cut out a particular cell in the tissue, but we also have the information on the staining markers called the features, like the staining intensity. And the first thing to approach the data is to, like, completely unbiased, reduce the data dimensionality on a principal and component analysis. And you can see that the true space, like central to portal vein area here, aligns very nice on principal component 1, which is the most important data separation dimension. Also, all known zonation markers were organized in the correct way, like ARG, ASOL, AS1, CPS1, high up close to the portal vein, and these two high up towards the central vein, and vice versa. So this all seems to work. In the paper, we then went a little bit further. We can show that we can actually see the main driver of this signaling, which is Wnt signaling, that originates at the central vein and propagates to the portal vein. This is why the most extreme changes are towards the central vein, because the signal quadratically decreases towards the portal vein. An effect that has also been seen by single-cell transcriptomics in this Nature paper from 2017. Overall, we find that about half of the liver proteome is space dependent, which is quite a lot. And now we're able to put back this information onto metabolic pathways. Here we bin the data, we bin the spaces into 20 bins from portal to central vein. We can see the urea cycle, for instance, very nicely divided from between portal and central vein. Most of it high up at the portal vein, parts of it high up at the central vein. Oxidative phosphorylation system in mitochondria also is space dependent, but observe here that it decreases linearly, most likely indicating that it is not driven by Wnt signaling. And we even uncover some new spatial biology, which is that peroxisomal very long-chain fatty acid metabolism is a central vein event. So if you take anything away from these 3 graphs, coconut oil is broken down at the central vein. So this is that was the part for the single cell DVP original methods paper. Now we are moving forward, and before I hand over to Caroline, I just really like to mention that proteomics is great also in a clinical setting. Clinics, especially pathology departments, work with formalin fixed and paraffin embedded tissues, or FFPE blocks, which are collected in tissue banks. It looks like this here. This is also probably with stylized. There are not that many blocks, but there are several millions of these FFPE blocks around the globe. And FFPE tissue is great for proteomics because it keeps the protein stable. So we can actually go back to these blocks that have been collected years ago, section them, and look at their biology with our methods. Here we are applying single cell DVP to the case of alpha-1 antitrypsin deficiency, which is a monogenic disorder affecting 1 in 2,000 people in Europe. Here you can see on these 3 stained sections that the protein alpha-1 accumulates in hepatocytes and leads to liver fibrosis. And the clinical heterogeneity is quite wide. While some patients will eventually develop liver fibrosis, others will never. And we're now driving a project using these technologies to understand this. Just a little glimpse in the data. We are also cutting out cells one at a time. Here you can see we're cutting out this one cell in the middle that has aggregates, surrounded by cells without aggregates. And we see a 30-fold difference in the levels of this alpha-1 protein in the cell in the middle compared to the outside, which is quite fantastic, meaning that the laser separation is extremely accurate. And what we now can do is make these spatial maps, a cellular cartography on FFPE tissue, this clinically relevant tissue. This is also work spearheaded by Sophia Steigerwald, I should say, another PhD student in the lab. So this is extremely laborious, I can tell you. We set a long time. And now I'd like to transition towards Caroline, because we have to ask now the question for the future. How do we select cells actually in a smart way? Do we continue picking randomly, or like everything as we did now in these two studies that I showed you? We are also having now thoughts, and we're actively developing methods to pick cells that look different, also using AI approaches here. So for instance, here on the right, you can see the morphology of these alpha-1 antitrypsin aggregates, which do look very much different in different cells. So here the underlying thought is that cells with different aggregate morphologies also have different biology. That is one way to group. But the problem is still, how do we know that we actually capture all aspects of biology? And now, going to Caroline, we now have a way to pick strategically much within a defined area, which we think is now going to make a huge difference in the future. Caroline.
All right, I'm taking over from here. So as you could see in the slide before, we really needed a new additional approach, which is not depending on different morphologies as step number 2. And this is especially important when we are focusing on one cell type, which we expect to change over space. So this would fully exploit the spatial potential of deep visual proteomics. So the main idea is to have an approach which is able to strategically select cells within a defined area. And what most people don't really see is selecting cells and back mapping their spatial character is one of the most labor-intensive steps within DVP. So there was a big interest in the automatization of this step. And that's basically when I joined the lab. So I came to the lab when the method paper was out and published. And I was trying to find my way through and was really interested in the single cell spatial proteomics field. And this automatization seemed to be really interesting and very much needed. So let me visualize the idea. We always start with high content microscopy. Within the image, we need to choose an area of interest. In this case, on this visualization down here, it's the area which is depicted. We can see we have a portal vein on the top and a central vein at the bottom. And then what we always do is we train a segmentation model based on deep learning. And this is just selecting one cell type at a time. So in this case, we can see that it was selecting hepatocytes. And then instead of randomly choosing cells and retrospectively define their spatial characteristics, we aim to beforehand define and choose cells of interest. And what we developed here is an approach which we call spatial mapping. So what we aim for in our case is we want to have an equal distribution of cells between the portal and the central vein. So we really want to cover the entire zonation trajectory. And you can imagine that this results in a really economic cutting, as we will only cut cells which will provide us with relevant biological information. So the idea of spatial mapping was born, and the potential of this approach is only valuable if this is done in an automated fashion. So on the next slide, I really want to point towards the most important part on the right, on the top right, which is Lucas and Paolo. So Lucas and Paolo are PhD and postdoc students in the lab of Carsten Borgwards. He's leading the machine learning department here at the MPI of biochemistry. And I really want to highlight that it's important to have interdisciplinary collaboration, because I think those are the most fruitful ones. So I try to motivate you to look around in your institute and see what kind of groups are around you, and if you can join forces in really tackling the big biological questions. So now I want to go through spatial mapping step by step. We're starting with what they call the information input, or what biologists would call a microscope image. We have a 4-channel image provided as an input. So we have the nuclei, the segmentation, and then as seen here, a marker for the central vein area in red and the portal vein area in orange. And the pipeline starts with a blurry algorithm. What they use is the nuclei image, and they blur the image and define black areas. This step requires quite some optimization. As you can imagine, that the blurry can have different degrees, as well as there is a threshold whether a black area is defined as a vein or as background. But once you have this, the black areas are defined as veins, and then depending on the portal and central vein markers, these black areas are defined or allocated as central or a portal association. The next step is the only manual step within the pipeline, which is the manual selection of the trajectory of interest, which is really easy because you only have to select point A and B. According to these points, A and B, the pipeline automatically segments cells surrounding the area of interest, and then further jumps towards the heart of the pipeline, which is a farthest first traversal algorithm. If we have some machine learning people among us, they're probably familiar with the algorithm. For all of the other people, I just want to summarize the approach in 2 sentences. What the algorithm requires is a geometric space. So we need to give it 2 points, in our case, a portal and a central vein, and an angle restraint. So what is the border on top and the bottom in this case, in which we still consider the area as relevant. And the algorithm starts with selecting one of the shapes in an arbitrary fashion. And the second shape is then selected in the maximum distance to shape number 1. And this is done in an iterative fashion, resulting in end shapes with the maximum distance from each other. And the end can be defined by the user. And you can imagine that this will result in an equally distributed set of shapes covering the maximal range of interest. For us, it's really important because now we can ensure that we really cover the entire zonation trajectory. And of course, we preserve the spatial characteristic, this time as a ratio value between 0 and 1. So 0 meaning being close to point A, okay, the central vein, and then 1 meaning it's close to the portal vein. And this indeed looks quite complicated, but we try to make this approach as user-friendly as possible. And all of this is now covered in the black box. So you, as a user, you don't really see all of this. The only thing you have to do is the selection of point A and B. But I still thought it's nice to get a glimpse into the black box. So with this in our hands, we could really increase the throughput dramatically, while having a more robust and precise spatial information. So now we can really move towards humans, we can move towards clinical cohorts. And the first clinical questions we were interested in was, is chemotherapy impacting the physiology of human liver? So time to scale up, and we have the clinical cohorts presented here. We have 12 healthy human and 6 chemotherapy-treated patients. It's really important to mention here that the chemotherapy-treated patients are not suffering from a liver-associated cancer, but a cancer of a different kind. Also, I want to highlight here that we are looking at the cohort including both sex as well as a broad age range. So the workflow is really adapted from the classical DBP workflow. We're starting off with sectioning our samples from the clinical cohort. We perform immunofluorescence staining and microscopy. This includes the spatial mapping pipeline. We isolate our single cells via laser microdissection, perform ultra high sensitivity mass spectrometry, and then data analysis, which I'm going to focus in a second. Of course, as a mass spectrometrist, I can't really go through the slide without mentioning a few mass spectrometry highlights. Clearly, the spatial mapping approach is pushing over throughput in the sample preparation side. However, recent developments in the mass spec world are dramatically increasing our sensitivity, namely the introduction of a novel instrument called Spatial. This is now allowing in-depth analysis of shingle shapes in a labor-free manner. Remember that Florian mentioned before, he used MDIA, multiplex DIA, to push the numbers and ensure a robust measurement. This is not required anymore, something that wouldn't have been thought to be possible just a few years ago. Also, developments in all kinds of areas around mass spectrometries are crucial. For instance, the chromatography. We are now really pushing the boundaries, and recent development allowed to double the throughput from 40 to 80 samples per day while still increasing the sensitivity. So we are in really exciting times right now. So now let me jump to the human results. The mentioned technological advances accumulated in the study quite tremendously, and we measured our clinical cohort in less than 10 days, despite the fact of having above 600 single cells. And we're receiving a median of above 2,500 proteins in just a third of an hepatocyte across our cohorts. Again, we can see the heterogeneity in our samples. As mentioned before, this is due to the area dependency. On the right, we color-coded the individual patients, and we can see that this is a shared phenomenon across patients. So we don't really expect a patient bias in the data analysis caused by this. Having a closer look at the identified proteins, here in this rank plot, we can see on the left the high abundant range. I marked some expected proteins, so some zonation markers, as we are interested in those in the study. So it's really good to see them as sort of a validation for us. But then going to the lower range, we can see transcription factors, which are really low expressed. So we can see we are really in the low sensitivity range. And in total, we could identify above 5,000 different proteins in our studies, which is a really good number to see. Going further, we wanted to see what is the overlay of common proteins within the healthy and within the chemotherapy-treated cohort. We expect chemotherapy to have an effect on liver physiology, but we of course don't expect it to change the entire proteome of the hepatocytes. So we're quite happy to see that chemotherapy seems to cause the loss as well as the gain of certain proteins, but sort of the core remains the same. This indicates a really robust measurement. We're still really interested in liver zonation just in healthy individuals. So we kind of focused on this cohort on that slide. So this is the really same principal component analysis which Florian showed. And again, we color-coded the shape mapping. So we can see the gradient shifting from yellow portal to black central. I really want to keep in mind that we're looking at 12 human patients with different sex and a broad range in age, as well as an unknown drug history. Despite this really strong clinical co-founders, we can still see a clear shift in the principal component analysis when color-coding the spatial positions in the sample. And this is also indicated on the right. So here we can see the principal component 1 value on the y-axis, and then the spatial mapping ratio between 0 and 1 on the x-axis. Again, we can see this really nice correlation between PC1 and the spatial mapping ratio. Taken together, we can again see that space is the main driver of the data variance in human hepatocytes along the zonation trajectory, and really see that this again indicates being the driver of the proteomic shift we can also see in mice. Of course, we can not only color code our PCA by the real space data, but also by the expression levels of different proteins of interest. Here we can see the PCA of one individual patient color coded by the expression levels of ASS1, which is a portal rate marker, and then on the bottom of CYP2E1, which is a central vein marker. And we can see that the directionality of this shift is vice versa. So this is really nice to see. But now, as we have preserved our spatial information, we can backlay this information on the original images. And of course, this will be really important once we have identified significant hits. And then we can really nicely illustrate these. Lastly, I want to focus on the zonation-dependent data analysis. And I want to bring back this nice illustration, a little bit small in the corner up here, which just shows liver zonation. And I want to go through the history of data analysis in liver zonation. And the most common and the oldest approach is to have 3 bins: the periportal cells, the midlobular cells, and the pericentral cells. Of course, we kind of lose a lot of spatial information, and we average over quite a bunch of cells, and it's quite a broad range of area. So what Florian did in his paper is he used 20 bins. As an average of 20 cells are within one trajectory. And this is sort of like a single cell analysis analog. But what we now try to do is to move away from this bin approach, because we kind of introduce arbitrary limits in here. And we want to move towards a continuous analysis. And the idea is to fit a linear mixed model through the protein expression. And this is in dependency of the spatial position of the cut shape. And with this, we're able to explore significant spatial expression gradients across our region of interest. And the significant hits are illustrated here in the volcano plot. I want to highlight that this is only the healthy patients, and we can see quite a number of significant hits approaching for both the central vein area and the portal vein area. Known zonation markers I labeled in black, so we can see we have some quite a number of additional interesting candidates, which you can hopefully soon see in our publication. And one aspect I really wanted to highlight as well is that mitochondrial proteins are known to be mostly found close to portal vein. This is a result of the oxygen gradient as well as the required metabolic functionality. We can see this nice cluster of OXPHOS protein down in the portal vein significant area illustrated in green. So this is again quite a nice positive validation of our novel approach. Of course, we also perform Go annotations of these significant hits, which result in expected annotations. So on the left, we see the central vein annotations. So here we can see hits for lipogenesis-related pathways as well as alcohol metabolism. And then on the right, for the portal vein, we do see mitochondrial and energy derivation-related pathways. So this is the work in progress, but we aim to hopefully soon publish this technology and provide a more in-depth data analysis, especially in the context of zonation patterns of healthy and chemo individually, and of course also of the comparison of the two. But for now, I kind of want to sum up, and we really hope that we introduced you to the concept of deep visual proteomics, which is merging microscopy and mass spectrometry. And I hope we could show you that it is now able to be performed not only on a single cell level, but on a single shape level. The approach is highly modular. It can be applied to FFPE in frozen tissue. And we now get deep biological insights with a median of above 2,500 proteins with just a third of an hepatocyte. We are able to preserve spatial information of single-cell proteomes and can back map all of the measured expression levels on the original imaging data. We are now able to have a strategic and automated selection of single cell, pushing our throughput and allow scalability. And finally, we're now able to move towards humans and clinical cohorts. Of course, this is not only work by Florian and me, it's quite a bunch of people, the entire Man Lab in our retreat in Venice here. Really great. Of course, we want to thank Matthias for not only great input, but also great instrumentation. The entire Man family, especially the deep visual proteomics family, but also our collaboration partners in the machine learning department, Paolo and Lucas, I mentioned them before, and also our clinical partners at the NIH. And of course, we're happy to answer any questions you may have.
Okay, we're opening the floor for questions from the audience as well as interview questions that Kristen and myself have for the speakers.
I can go ahead and kick off with the questions. First of all, thank you so much, Florian and Caroline. Those were great talks. And this question's for both of you. So when did you know that you want to focus your career in this area of research?
Caroline, go ahead.
You want to talk?
No, go ahead.
No, I personally decided right after my master thesis. I've been working on basic biology beforehand, a lot of cell biology, and I really wanted to get towards the clinical aspect. And I do find mass spectrometry is just at a rise, and it's really powerful, and it's a good time to jump on and really see all the developments. I mean, I'm in my second year of my PhD now, and I'm already in the third generation of mass spec. So it's incredibly fast moving, and this was really what brought me into the lab, and it's also quite a nice lab.
Yes, it is. Thank you, Caroline. What about you, Florian?
Yeah, I did my PhD in mitochondrial biology on the Drosophila genetics, so mostly crossing flies. But at some point we got into transcriptomics and proteomics. I did the data analysis basically for the whole lab, and always had a little bit of liking of computers, programming, I'd say. Then when I was done with my PhD, I thought, now I have to learn now proteomics properly. So I went to Matthias Mann's lab, which is, I guess, one of the best places where you can be if you want to learn properly. And it is. I'm having a fantastic time here. Single cell and spatial proteomics is certainly the forefront. So it's both very hard to avoid, but it's also super exciting if you want to live up, or if you want to be creative and shape novel technologies out there. And yeah, I absolutely love it.
Great.
I think it's interesting that you had this slide showing how your team is broken out among the different organ systems. And so is there ever any, plans for some kind of synergy and characterizing of, maybe the same types of cells, but from different organs? I'm sure you're doing things like that. Can you speak to that at all? No.
No, it's a good guess. Yeah, we're working on this. Shouldn't mention too much, I guess.
Yeah, no problem. One of the things I wondered was when you're working with these different teams with different expertises, do you now have a way that you start communicating with them? And is it different? For instance, for myself, I find it different talking to a pathologist versus a biologist, and I try to incorporate tips such as that for my trainees. So I just wondered if you had any points, maybe Florian or Carolyn.
Yeah, so within the lab, we are quite a heterogeneous group of people. The lab's quite big. So in Munich, we are more than 40 people. Some of them are good on the bioinformatics side, others are good on the machines. Then there's molecular biologists, and then there's also the clinicians. So you do get this training on a daily basis. We have interest groups here. For instance, there's a deep visual proteomics interest group where we just meet and discuss everything basically. But of course, many of our projects are highly collaborative. So we talk to clinicians a lot. And a lesson I've learned is go out there to medical conferences, really, which are completely out of your comfort zone. And you realize that, first of all, there are many smart people out there, but that everyone's curious in what you're doing. I really love that. And this is the way how you learn to communicate with each other.
That's so important. Thank you.
Great. And I'll take the next question. So you guys already showed, scientific reasons why the single cell deep visual proteomics is so important. But maybe in your own words, can you answer, why is DVP important for understanding biological heterogeneity? What benefits do you think you achieve with the spatial analysis, and is it worth the work? So more of a theoretical question.
I do think if we really want to answer biological questions in depth, we really have to go single cell. I mean, there's this nice cartoon of a fruit salad and a smoothie. And when you analyze this movie, which is kind of bulk analysis, you can never say what is causing the bad taste. Was it the bad banana or the bad apple? And this is extremely important if you move towards disease, such as cancer. And I think this is where really the spatial characteristic is getting important. And then if we move towards DVP, I mean, DVP is among the only technique which can analyze in the proteomics field in physiological tissue. So we really have intact tissue, we image them, and then we isolate the cells compared to, for instance, FACS, where you like really dissociate your cells within the tissue. So I think this is really the powerful method. And then proteomics, of course, proteins are the functionality of the cells. And we do slowly move towards sensitivity in which we can even go towards post-translational modification. This is a layer of information that transcriptomics could never reach.
Thank you, Caroline. Fantastic answer.
Florian, do you have any comments on that?
Oh, well, just adding to this, we see on a daily basis that bulk measurements are not good enough for the tricky biomedical cases. You will not understand why a particular tissue appears pathological or sick over another in many cases. So this cell type enrichment just allows the biology to emerge much clearer. And laser microdissection is one important tool to do this because it conserves the spatial information which we need. You need to know in which environment is a cell, what nutrients does it have, what's the oxygen, what are its neighbors, with whom does it communicate. This is absolutely crucial to understand how a tissue works and how it gets sick.
I agree with you. Thank you.
That being said, I think there's so much potential to, or let's say there are so many problems actually, because it's such an enormous amount of data. I think we're all struggling with understanding this. So for the students out there, there is years, if not decades of work ahead of us, really.
Yeah, and then using it with artificial intelligence is a whole nother story. But I do think that right now, the data that we're building is actually important for people to use in modeling by artificial intelligence. That was one of the things that kind of came out at our mass spectrometry imaging conference in the Americas recently. So very important work. I have some questions on the experimental details, although you have them in the paper. Carolyn, you had mentioned, you had briefly touched on the chromatography that you were using, and I was really curious about that. Do you know any of the details as far as like the method development that went into doing chromatography on these low abundant cells and proteins?
I mean, one of the key like developments which has been established.
So like as far as like testing like types of columns, column diameter, like can you speak to anything that made the biggest difference?
I think the biggest impact was really the low rates, the low flow rates.
Okay.
The main development, I would say.
Yeah, I like that you called it something like the whisper flow rate. It's just so low. And then the other question I have is if we're trying to take these and adapt them to clinical platform, we really have to speed up this type of analysis. And I wondered if you had done any work as far as speeding up the chromatography of these very small samples. So maybe I'm asking the wrong person.
We're using the EvoSep system quite a lot, so this comes basically out of the box to us. But so I'm now speaking for the developments that are out there on the market. I think now we're starting to have viable approaches for 120 samples per day, and this is being scaled further. The question is, though, whether we can ever come to a throughput where we are biologically satisfied, or if you have to tackle this project or this problem in a different way, like what Caroline was alluding to. You don't have to measure everything. You have to measure the right cells, really. And I think there are a couple of steps before you throw everything on your mass spec where you can make the right decisions. So maybe having 1,000 cells, or let's say 5,000 cells for a clinical cohort are enough. I don't know. We'll see.
Do you think that using like conventional mass spectrometry imaging, perhaps by multi-imaging, might be a good approach to like a first pass? A lot of analytical strategies will, particularly for clinical, will have like your first pass, and then they'll actually isolate the special cases, which will then go into a detailed analysis.
Yeah, absolutely. I think this is a great point that you're making. These multi-omics approaches here, like first choosing one that is fast and then going in depth. The spatial mass spectrometry based proteomics is slow. It's intrinsically slow. We know that, but it's precise.
Yes,
so
it's very detailed on the sequencing and the PTMs, right?
Exactly. So you have to ask the question when you really need it. If it's the case for novel biomedical discovery, I think there's no way around it.
Okay, so if you were leading a new study on the same topic, is there anything that you would do differently?
Caroline, you start.
I guess you learn a lot along the way. I mean, especially for me, I studied a PhD. I guess you just learn a lot just by default. I guess it's, for me, so this entire automatic approach came along like on the way. It didn't start with it, and then we first did a pilot, then the automatic approach. So I think like talking to people and get expertise from different fields is really powerful. So I think I would have done this maybe a bit earlier. But apart from this, I think we're really happy with how it went. Of course, you learn like small things, but the mass spec world is so fastly like developing. So if you now, probably in the moment we talk, there's a new gradient being developed. So I don't think I wouldn't change much.
I think this is really the issue. 1 week you're so happy with your 3,000 proteins that you've quantified, and then the next week someone's better. So the field is turning around so fast. It's very difficult to afterwards say like I would have done this or that better, because things just fall into place somehow, which makes it both exciting but also sometimes frustrating.
Yep. Yeah, it's an exciting time to be a mass spectrometrist with the instrument developments, the method developments, and you guys are definitely on the cutting edge of it.
So Florian and Carolyn, how do you think this method would apply to a fibrotic liver tissue section?
Liver fibrosis is actually one of the reasons why I came to this lab. Extracellular matrix is fair game for mass spectrometry-based proteomics and very difficult for transcriptomics, right? Because the extracellular matrix is just so stable. What happens inside the cells that generate this matrix on a transcriptomic level just does not represent what's inside the extracellular matrix. Also, we know that there are so many post-translation modifications that stabilize these scars or make them more vulnerable. So we are really thinking that yes, laser microresection with smartly choosing the areas, the fibrotic areas, can be a key to understanding how a fibrotic scar can be broken down, how you can trigger this. But also there is a number of technological steps still missing, especially what Caroline said at some point. Measuring post-translation modifications at this low input level is extremely tricky. We are getting there, but I think this is one step that is still needed.
Yeah, and especially when you add that they're from formalin-fixed paraffin-embedded tissue.
Yeah, well, there are literally millions of samples out there that you can analyze. But this is the great part. So FFPE samples are usually organized by pathologists or clinical boards, and to get access to them, you have to talk to these people and design experiments in the best possible way. You learn a lot on the medical side, but also your experiment just gets much better because you're not only sitting and reading, but you get input from people who actually work with these diseases on a daily basis.
I found it's useful to ask the pathologist, what are you interested in? And they don't, the pathologist, at least, the clinicians will frequently give you a lot of ideas, but the pathologist in particular, you know, they're the ones that have these weird cases of different case studies that they have in the back of their mind, like, well, I'm seeing a lot of mucinous triple negative breast cancer. I had that from a pathologist recently.
Yeah. Pathologists are such a biologically intelligence. No AI can keep up with them at the moment.
All right. Are there any more questions? Do you guys have anything else to say or even questions to ask us? Nope.
Well, where do you think is spatial mass spectrometry-based proteomics going? What's your input?
So for me, I do think that it's moving toward the ability to predict disease and not just predict you know, where there's a huge tumor, like in the picture behind my head, there's a tumor right there, but able to predict it down to a single cell. So and there's also cancer field effects, and those actually do get taken up by the extracellular microenvironment. But I think my concluding statement for this session would be that I think both the fields of conventional mass spectrometry imaging, using, for instance, using MALDI or DESI, that's Kristen and my background, we need the proteomics field and we need those developments and we need to work together synergistically because that's how we're going to be able to solve disease and be able to detect it at the single cell level.
Yeah, I completely agree. You know, single cell or regional specificity for figuring out metabolites, lipids, and proteins. That's where we're going to start to really understand diseases or biological processes at a mechanistic level, which we've always been trying, but I think we're actually getting there now with the depth of coverage and these new advances that everyone's making and the new instrumentation. We can start to achieve those.
Great. Well, I want to thank you both again. Thank you so much for putting together the talk, and thank you for giving such a great presentation that's a recorded resource for our students and attendees at the US HUPO. So thank you.
Yeah,
Thank you
thank
very
you
much.
guys.
Thank you.